Well, hello everyone. I'm Rick Wilson, the Director of Scientia, and I'd like to thank you for attending today's lecture. This year's series is entitled PANOPLY, and I take the meaning to be a broad tapestry of talent. And the aim of the series is to showcase the many different streams of research and ideas here at Rice. Scientia members hope that this will encourage a good deal of cross-school fertilization. Today's general topic is animal behavior. These lectures address the causes of animal behavior, including humans. Oftentimes, debates on this topic focus on nature versus nurture. And our three speakers today actively work on the topic of behavior and will weigh in as to whether such a simple dichotomy even makes sense. Our first speaker is going to be Julia Saltz, an assistant professor of ecology and evolutionary biology here at Rice. She received her AB from Princeton University and her PhD in population biology from UC Davis. She was a postdoc in the molecular and computational biology program at USC. She joined Rice in 2014. She's received numerous grants from the National Science Foundation, and her publications appear in The American Naturalist, Ecology and Evolution, and Heredity, among many other journals. Our second speaker is Caleb Kemere, who's an associate professor of electrical and computer engineering at Rice and an associate professor of neuroscience at Baylor College of Medicine. He received his PhD from Stanford, was a postdoc fellow at the Keck Center for Integrative Neurosciences at UC San Francisco before joining the Rice faculty in 2011. He's a winner of a 2013 NSF Career Award, and in 2014 received a 5-year grant from the National Science Foundation to study deep brain stimulation. As well as a 2015 recipient of Brain and Eager Award from NSF, and as a co-recipient of a Neural Engineering IGERT grant. Our third speaker is Simon Fischer-Baum, an assistant professor of psychological sciences. He received his BA in neuroscience and behavior from Columbia, and his PhD in cognitive science at Johns Hopkins in 2010. He was a Beckman Institute postdoc fellow and joined Rice in 2013. So from my standpoint, we have a group of youngsters here today to address us, but I think that's only from my perspective. His work has appeared in numerous journals, including Cognition, Frontiers in Human Neuroscience, and Psychological Sciences, among many others. I am very much looking forward to these three talks. I think they'll be enlightening, and I turn the mic over to the first speaker, Julia Saltz.
Thank you all for coming. Thank you for that lovely introduction. I'm Julia Saltz, as he said, and today we're going to be talking about determinism in behavior. And so I chose this topic because, as scientists, but also as members of a society, the reasons for our behavior and often the reasons for other people's behavior are incredibly important to how we live our lives. So we can think about both our actions towards other individuals, both people in our families and just other individuals that we encounter, and also laws and policies that we make as essentially resting on hypotheses about the causes of behavior. And you can even think about, back to Shakespeare, and I'm sure much before, musing about our faults being not in our stars, but in ourselves. And now NASA has conducted an experiment to test this hypothesis. So we have these two identical twins, where one of them went to the International Space Station for a year, and the other one didn't. So this is a powerful approach to try to test the effects of environments on individuals who have the same genotypes. And so what I hope that you'll see today is different scientists, colleagues at Rice, who come from different intellectual backgrounds and work actively on this issue. And so hopefully we can share a little bit about what we've been finding in our different respective fields. And what I want to emphasize is that we're not going to resolve questions of what makes us who we are and can that be changed over time in the next 45 minutes. So I'm sorry if you came expecting that. You will be disappointed. But hopefully, again, you can get a flavor for the types of scientific research that we're conducting on these topics and also the opportunity to learn more in the future. Okay, so I'm an evolutionary biologist, And so in my lab, we investigate the causes of behavior, focusing on an organism that you might not think of as your first one in terms of answering questions about what makes us who we are, and that is fruit flies. So fruit flies may have been inflicted on you at some point in your education or also at some point in your kitchen. And so you may think of them as annoying or boring, but actually they're just like any other animal with complex ecological and social lives that have evolved over thousands or millions of years. And the beautiful thing about flies from our perspective is that we can use them to conduct what I think of as the ultimate twin study. So just like these identical twin astronauts that have the same genotype but different amounts of going into space, we can have fruit flies with identical genotypes, but instead of just having 2 of them, we can have hundreds of individuals who are genetically identical or genetically different. And of course, because they're not humans and instead are flies, we can manipulate the environments that they experience. So we can compare in a very robust way individuals who are the same or different in their DNA sequence, their genotype, and individuals who are the same and different in their environment to try to understand how those different factors impact their behavior. So today what I'd like to do is share with you some recent findings from my lab that, in my opinion, dispel some of the most common misconceptions about the way that genes affect our behavior. So the first one I want to talk about is this idea that I see represented in popular culture that because our genes remain the same over our lifetimes, any behaviors that are influenced by genes must also be the same. We can't change them. And then we'll talk about whether behaviors are genetic or learned, and then we'll get to what I think is the sort of heart of these misconceptions, which is this dichotomy between nature and nurture, this false dichotomy. Okay, and so the reason that I don't agree, and I think it's a misconception to say that because our DNA sequence remains the same, we're trapped in the behaviors that our genomes lay out for us, is that although it's true that our DNA sequences remain relatively consistent throughout our lives, the effects of our genes on our behaviors can change rapidly and dramatically. And so to illustrate this, I want to introduce fruit flies showing aggression towards each other. Okay, so we can see these fruit flies fighting each other. So here, these are our little flies that we measure in our lab, and we paint tiny dots on them so we can tell which ones are which. Okay, and so aggression in flies, and there's also some research on aggression in human, has a genetic basis through any sort of meaningful description of that term, sort of at the level of both individuals and species. So we know that flies can do aggression the first time they see another adult male. Males with different genotypes show different levels of aggression, and other labs have traced the neurogenetic basis of aggression to things like dopamine receptors in the brain. Okay, so this is a genetic behavior if there ever was one, which I would argue there isn't. But we can also use the power of our twin studies to examine fruit fly males that have identical genotypes, just like our identical twin astronauts, but when fighting against different opponents. And we often see that males who are genetically the same will be highly aggressive towards one opponent, but not very aggressive at all towards a different opponent. And this is both incredibly obvious, but also kind of important. Because of course individuals don't behave the same regardless of what's going on around them. Everyone adjusts their behavior to their social and ecological situation in a wide variety of circumstances, and yet our genomes stay the same. So you can think about the role of the environment in shaping the relationships between our genotype, which is stable, and our environment, which is ever-changing. And so next time you put on your glasses, you can think about yourself as subverting the relationship between your genotype and your behavior by changing the environment in a way that improves your vision. Okay, it's as simple as putting on glasses. Okay, so another common misconception about behavior genetics, which I encounter a lot, again, even among very smart and lovely people, is that behaviors can be categorized as either genetic or learned. And there are many reasons that this viewpoint is not consistent with evidence. Hopefully you started to think about some when I was talking about aggression. But my favorite example of this is that learning itself can have a genetic basis and differ among genotypes. So here's data from an experiment where we asked flies to choose between two different types of fruits. Think about apple and pear, or whatever your two favorite types of fruits are. And then we put poison in one of the fruits to make that fruit bad. And we can measure how well flies are able to identify, aha, now pears are poisonous, and so I shouldn't eat them. And this is data from 116 genotypes. Each of these bars is a different genotype, and this y-axis is a measure of how well flies perform on this simple cognitive task. And so over here we have our individuals who are pretty good at learning what foods to avoid, and here are our bad learners and everything in between. So as someone who's interested in studying natural genetic variation in behavior, this is like my favorite slide that I've produced in my career so far. And it again highlights that learning itself can differ among genotypes. And we're now extending this study across longer evolutionary timescales using a natural experiment that evolution has performed for us. So there's a special, unique kind of fruit fly, Drosophila seychellia, that eats this fruit, Morinda citrifolia, also called noni fruit, that is poisonous to all other flies, even its closely related congener, Drosophila seychellia. And so what we've been studying about these flies is how this evolutionary shift onto this previously poisonous fruit has influenced their learning. And so far we're finding that this shift has been accompanied by an evolutionary improvement in learning preliminarily. And so we're still kind of putting the pieces together about how these changes in the environment can lead to changes in learning, but this again underscores that learning itself can have a genetic basis and evolve. Okay, so finally let's get to I think the most important misconception about the role of genes in shaping behavior, and that's that there are two factors that can influence behavior, nature or nurture. And we always, when we see these, the words nature and the words nurture, there's always this versus in the middle, like they're fighting for dominance. And so we've already seen an example here, and I'll give you another one in a second, of how dichotomizing the causes of behavior into these categories can be misleading or even just impossible, right? So for example, we have genetic differences in whether flies choose which environments they experience, and we also know that in these flies that eat the poisonous fruit, they have experienced an evolutionarily accelerated loss of taste and smell receptors, which is actually similar to humans. We are also rapidly de-evolving smell receptors. And so we can think about the environment as shaping not only animals' immediate experience of the environment, but also over evolutionary time, how they're able to perceive the environment in the first place. So the idea is that these flies have shifted onto this toxic fruit because they can no longer smell how bad it smells and tastes. We also see, we do quite a lot of research in my lab on looking at genetic variation in the environments that individuals choose to experience in the first place. And so I already told you about some of our work on choosing different fruit types, but we also see this for social environment. These flies are moving around and we give them the opportunity to choose which individuals they want to associate with. And what we see consistently is that some of our genotypes end up associating with a lot of different flies, which we call a large social group, and other genotypes end up in a smaller social group. And so this indicates that genetic variation can influence which environments are experienced by particular individuals. So instead of thinking about the role of nature and the role of nurture, we can think about how genetic variation in behavior influences the environments that individuals experience, which you can think of as kind of nature via nurture. And then these environments, both within individuals' lifetimes and also over evolutionary time, influence the relationship between genes and behavior, which you can think about as the opposite thing. And this endless cycle is what drives us to develop the behaviors that we do during our lifetime, and also evolutionary changes in behavior. And this plot essentially summarizes why I think the nature-nurture dichotomy is unhelpful. And with that, I'm happy to turn the mic over to Caleb.
I am Kemere, a professor here in electrical and computer engineering. And I also have an adjunct appointment at Baylor College of Medicine in their neuroscience program. And so I like to think of myself as a neuroscientist. I think that what's great is that once you get tenure, you can decide who you want to be, and nobody is the boss of you anymore. So I am a neuroscientist, and I am representing today. I'm going to start with my acknowledgement slide, just in case I run out of time. I think Rick has a bell. So I'm going to present some data that one of my very good graduate students, Shaktara, gathered for me, and videos that he made very nicely. And I also want to thank all the people that gave us money. So animal behavior is a huge topic, probably hard to cover in a year, let alone 45 minutes. And so we, as you've already figured out, decided that we were going to answer a slightly simpler question of how nature and nurture are affecting animal behavior. And because I study learning and memory, I thought I would further refine this question into basically just a bunch of little factoids that I'm going to share with you. But this is the theme of those factoids, which is that our brain is pre-wired to want to learn and to want to remember. And that's probably one of the organizing principles of my life and probably your life. There's a lot of stuff that happens to us, some of it random, some of it intentional, some of it based on decisions that we make, some of it based on decisions that other people make, that affects how we learn, what we remember, and how our brain is working. So nature is the circuits that we're born with that allow us to learn and remember. Nurture is all the stuff that messes with them. All right, so I want to focus on something interesting. And that thing that is interesting is curiosity. So I feel like that's very meta. This is a picture of my daughter, Lydia. She, in this picture, I think is about 4 months old. And I want to point out that infants at this age, 3 to 4 months old, already show a strong preference for new things. So you can, and people do, though not me, fun experiments where you have like the old toy and the new toy, and even before they're like good enough to like grab the toy, they'll like look at it, the new one, as opposed to the old one. So why is it that why is it that happens? Well, there's sort of two answers. So one answer is that when you're in a new place, when you're offered something new, your brain goes into a very interesting state where all sorts of cool neuromodulators, even hormones start activating complex circuits. But then the other question of why that happens is presumably this is critical for us to learn. I use the word. It's critical for us to be able to become more fully human, and also in order to survive, to learn about our environment and to enjoy it. So here's a complex circuit example. This is the circuit that is involved in just the detection of novelty. So even knowing that something is new requires your brain to do some effort, right? You have to look at your environment. You have to recognize that something has changed. And then that excites your brain, and it has this really nice positive feedback process. But one of the principles of this diagram is that there's a part of your brain called your hippocampus, which I'm particularly interested in, which plays a key role. So if you're taking away things from this section of the talk, there's pieces of your brain that are really important, not just to learn and remember, but even to be curious. So how do we study curiosity in rodents? So this is a sped up video. It's like 10x speed. And this is a rat, and he's in a box. And he's not learning anything, or we're not asking him to learn anything, meaning we're not going to reward or punish him based on whether or not he does what we want him to do. The thing that has happened is that a few minutes earlier, he'd been put in this box, and this Tupperware, I'm sorry about the quality of this video, this Tupperware was over here. And so what happens is that he goes into this box, and he sniffs around. Let's play it again. And he sniffs the Tupperwares. He explores. And we can quantify, this is some quantification, just focus on the blue bars for now, that he actually spends quite a bit more time sniffing this box than he does this box. But if you just look at this is a rat, lives in a cage. We put him in this big meter large squared thing. And what does he do? He like looks around. He's rearing up on the sides. He's like, how big are the walls? In my mind, I imagine that he's looking for food, but he could also equally be looking for predators. But he's exploring. It's interesting. It's new. Now, what's interesting about this is it's not actually new. He's been in this box many times, but every time he thinks, well, maybe something is new. And so what's cool is this is a neural circuit that's just driving behavior. It's driving learning. And the thing that we can see is that even without being rewarded, we can see that there is a memory that is formed. So curiosity and memory are intimately tied. So in this case, he explored the box 5 minutes previously. He took a short nap. He came back in, and then he remembered that the location of this Tupperware was in a different place. All right, so this is there in your brain. Rats do it. We don't have to encourage them to do this. This is a really nice way that we can just study these endogenous circuits, so we can study nature. So now let's ask, what affects the ability or the capability of our brain to do these sorts of behaviors? Okay, so curiosity requires memory, so it requires us to remember that the way that the world is in order to understand what's new. It also requires us to explore, right? So we can't actually find out something about the environment if we don't explore it. And then I'm going to talk about how there's a lot of loops that feed those things back on each other. All right, so another good takeaway. Things that happen to us, for example, if we sleep, improve our memory. They allow us to be curious. And so we can say the opposite of that as well, which is that if we sleep deprive people, if we sleep deprive rats, then they become less curious. And I want to show two things about this. First of all, if you, these rats haven't been sleep deprived that badly, just 4 hours. If you sleep deprive some rats, then their ability to do this task decreases. So in both of these sleep deprived groups, their behavior is slightly decreased, but immediately right after the sleep deprivation, they're showing basically no preference. They don't notice that anything about the environment has changed. But part of that is being mediated by the fact that they're also exploring a little bit less. They're tired. They're not running around, right? So we immediately see that things that affect neural circuits of learning, like sleep deprivation, act in multiple ways, right? They act directly on the brain by affecting neuromodulators, by affecting stress hormones, but they also act on our body. And there's this loop that means that if we can't actually crawl around our environment and look at it because we're exhausted, then we learn less. So it's interesting. So, okay, already we see that the effects of nurture on the brain circuits are a little bit complicated. Now, these things can actually be more profound. So for example, if you allow rats access to a running wheel, you find that in their hippocampus, many new neurons are born. So one of the things that's really cool about the hippocampus is it's the only part of the brain where you get new neurons throughout adulthood. The stress hormones go down. The animals explore more. Conversely, if you cause a rat to become depressed, then all of those things will decrease. Exploration will decrease, memory will decrease, neurogenesis will decrease. So there's a number of factors that will affect our ability to form new memories. Now, some of those, as I already described, affect our desire or ability to explore. Other things like anxiety or stress will also reduce exploration. So actually, a classic test of anxiety is to put an animal into an environment where there's an exposed section of the maze. And animals that are anxious will not explore the exposed section of the maze because they're anxious. So you can't find out something new about the environment if you don't explore it. And all of those things are working. They're working to affect how we behave. And if we're stressed and so we don't exercise, then there's this positive, negative feedback loop, this feedback loop that's working on our brain, that's causing our behavior to change, which is working on our brain, which is causing our behavior to change. But there's also positive feedback loop. loops. So there's a very large literature reaching back about 30 years where they discovered that some mother rats randomly would lick their infants more or less. And so some creative investigators looked at those baby rats and discovered that ones that had been licked more actually ended up being more curious and having better memories later on in their lifetime. They also explored more. Turns out the ones that licked less perform better on tasks where they have to learn how not to get shocked, for example. So there are effects of upbringing, so other people acting on the rat change how they behave, and then also even just housing rats together in a way that they can be social with each other improves their memory. So learning is not only just based on curiosity. It's not just exploring the environment and seeing what's going on there, right? The most interesting, the most critical parts of learning are ones where we use feedback from our environment to change our behavior. So we can sort of split that kind of learning into two pieces. So one is learning based on reward, and one is learning based on punishment. So this is just an example of tasks that we do in my lab where this rat has to learn that in this maze that he will only get milk in these reward wells if he alternates. So basically he has to do this outside arm to middle, the outside arm back to middle, and so on pattern over and over again. If he does that correctly, then he gets a lot of chocolate milk. He's happy. If he does it incorrectly, he doesn't get the chocolate milk. So what's interesting about this, right, is that in order to learn this task, we can't tell the rat, you must alternate. The way that they learn to do the tasks is that they explore their environment. They get rewarded, maybe seemingly randomly to them, and they put together those rewards, the pattern of rewards, in order, and that allows them to learn to do this behavior properly. And what's cool is that we can see that it turns out that they learn two phases of the task at different time frames. So the phase of the task, which is that if they've been in an outside arm and they return to the middle, they will get reward in the middle. They learn after about 2 days or about maybe 2 hours of being engaged in this task. Whereas the part where they actually have to alternate takes them a little bit longer, probably 4 or 5 days. And then some of these, this is just data where we've been studying in my lab the actual circuits, the specific ways that this learning happens. And we find that there's specific circuits that are using memory in special ways to allow this kind of learning. All right, so when we think about reward, okay, so this is again a circuit, reward-based learning, that dominates large parts of the neocortex, and it involves the hippocampus. Now there's aspects of that circuit that are a little bit weird and that can be affected by our interaction with the world. So one of those, for example, is that if you give reward randomly, then it turns out that you can actually get rats to do more trials. So we like this because we don't want them to be full, because then they'll stop working and they'll just start sniffing around the maze because they become curious. But those same brain circuits can get twisted or maybe used incorrectly for things like slot machines. I don't know if you've ever thought about this. Why do people play slot machines? So this neural circuit that's saying, I will take reward and I will try to make a pattern out of it, gets hijacked. And for some people who become addicted to slot machines, I think anybody who's playing a slot machine probably is being unwise. And as a result, they keep pushing this button, even though the reward is actually just being randomly delivered. I imagine that the way that works in our brain is that there's a little person inside the brain that's saying, well, I know I'll figure out the pattern. I know that I can figure this out. Yes, I know it seems random, but I'm going to figure it out. So I'm going to keep pushing the button because I will eventually get rewarded a large amount. Substances of addiction, especially things that act on the dopamine system, strongly hijack this reward system. All right, I want to show you one more thing and then we'll conclude. And that has to do with learning when something bad happens to you. All right, so this is a task that I had a colleague at Baylor College of Medicine and I collaborated on. So his student had this great idea. So I mentioned that one of the tests of anxiety is that you put an animal into a maze where there's portions that are dark and portions that are light. And if you do this with a rat, this is what will happen. So you put them in, very first time they've experienced this sort of environment, and they hang out in the dark part of the maze. This is just a linear track that looks something like this. And then they'll walk over into the light part of the maze, and then they'll turn around and they'll run back into the dark part. And they'll walk over into the light part of the maze and they'll run back. run back into the dark part. And they'll say, well, let me go see what's over there. And then they'll run back. And they sort of do this, and eventually, they end up with a bias towards the dark side of the maze, but they've explored the light side. Okay, so now, for this experiment, we did something that was sort of neat. So the second day that they explored this maze, we put them down in the dark part of the maze and shocked them. So we gave them a negative signal, a signal that they needed to learn from. Something bad happened. So how do they react? Well, so now the next time they're put in the maze, they run over to the light side of the maze, and now they come, and their behavior is effectively inverted. So they have this natural bias to preferring dark and high walls, and now they're preferring the part that's brightly lit. And what we actually find, this is a really cool paper that if you're interested in this stuff, we can talk about more afterwards. We find these neural sequences that look like every time they turn around, they're actually remembering being shocked. It's really cool. But the point is that we have neural circuits that allow us to learn very rapidly. One time, this rat got shocked. And for the next 20 minutes, 30 minutes, 40 minutes, hour, they were smart enough to say, okay, I'm going to avoid that part of the maze where I got shocked because that was bad. All right, so I want to say two things about this. First of all, if you do this a lot, so if you get a lot of negative feedback to an animal, and especially if you do it randomly in a way that they can't figure out the pattern, that is called chronic mild stress, and that is a way to create a depression phenotype. So when people want to study depressed animals, the way that they do it is that they mess with them a lot in random ways. So when we think about people that we know that may be depressed, it may be that their neural circuits that are reacting to the world have been entrained in this way. So one of the things that's cool about that, or interesting about that, is that all this negative learning doesn't last forever. So this rat in this maze, if you put him back the next day, it's actually the problem with this experiment, was that we were going to study this stuff further and further on. What we find is that after a day or two, the rat's back to liking the dark side, because we've only shocked him that one time. They're like, well, that was just bad luck, and I should just get on with my life. And so it's very interesting to think about how the way that we interpret what happens to us, the way that we create models of the world, the way that our memory interacts with our experience will then affect how we go forward in life. And I think that resilience, so the ability to have negative things and be able to react positively to them is something that can be learned. And it's something that can be rewarded. And I guess that's what I want to conclude with. So we're pre-wired with these neural circuits that make us curious, that allow us to learn from reward, that allow us to learn from punishment. What happens to us, that reward, the punishment, the patterns of exploration, can affect those neural circuits. They can have a huge impact. But I think that we can teach our brain to react well to the world, and maybe more profoundly, and hopefully this might be something that we discuss afterwards, we are social animals. We are providing feedback to other people. We are providing meta-learning signals that allow other people to learn. And so if you take nothing away from my talk except for this, my encouragement would be that if you're thinking about nature versus nurture, think about the fact that you have the ability to nurture other people. You have the ability to change how their brains are working. You have the ability to affect the outcome of their life, both in the short term and the long term. So that's the main takeaway. All right, awesome, thank you. Do you want to print?
So I'm a psychological scientist, but I'm also a neuroscientist. I don't have tenure yet, so I still describe myself as a psychological scientist, I guess. But I study humans, and so I just want to orient you to this. This is a brain. This is a human brain.
Humans in the dark side and the light side.
We don't shock them. Or really get to control their experience at all when they come into the lab. So this is a human brain. It's the organ that gives rise to all of your behaviors, save the spare involuntary reflex. It's also responsible for your thoughts, your emotions, your feelings, your sense of individuality, your sense of identity. So when we're talking about determinism and behavior, we should also think about how the brain works. and the ways in which it develops, and how the ways that it is structured and organized is governed both by genetics and by experience. So in this vein, it's remarkable to reflect on how similar the structural organization of the brain is across people that vary drastically in cultural backgrounds or experiences of the world. So across all sighted people, the back of the brain is the part of the brain that processes visual stimuli. Across all hearing people, the top of the temporal lobe is the part of the brain that is responsible for audition. Our ability to sense touch and our ability to move, our bodies are governed by neurons that reside in almost exactly the same location across all humans. And even within a single system, there is really strict organization. So if you were to just tell me the location of a firing neuron in the part of the cortex that's responsible for early vision, I could tell you with reasonable certainty exactly where in the visual field the light was present. If you give me a scan of somebody's brain, I can identify a little knob on there that I know if I stimulate it, I can get your finger to twitch involuntarily. At least at the level of our sensory systems, there seems to be remarkable consistency across people in how the brain is structured. And this consistency goes beyond basic sensations. So for example, across people, across cultures, there seems to be specialization within the visual system, with different parts of the brain responsible for processing different kinds of visual experiences. A part for processing bodies, a part for processing objects, a part for processing places, and a part for processing faces. And if you have a stroke that damages this part for processing faces, you're left with prosopagnosia or face blindness, which is a selective problem in either perceiving or in recognizing individual faces without other corresponding visual impairments. So the organization of the brain is remarkably consistent across people and across cultures, despite all of this difference in their background and experiences. So because of this, you might want to assume that this is somehow genetically determined. In fact, Khaled sort of suggested this when he said that we were hardwired with these circuits. I'm going to argue that we are not. But of course, cognitive neuroscientists like to talk about things in evolutionary terms. When we talk about the face area, we like to imagine ourselves back in the Serengeti and how, as social animals, it was really important for us to evolve a specialized circuit for recognizing faces. But of course, it's more complicated than that. If anything, if you take anything away from all of our talks, is that it's just more complicated than that. And so I'm going to talk to you about an area that I research on, which is an area where we appear to have specialized circuits, and yet we could not have possibly evolved to have these specialized circuits. And so I'm going to introduce this as the greatest technology in human history, since I'm in the engineering building. It's a technology that connects people across time and space. It allows for the accumulation of our intellectual achievement, and it is a lossless external memory storage device. It is, of course, the written word. The written word evolved about 6,000 years ago, which is not nearly long enough for it to be something that our brains have evolved to do. And so we're left with a paradox. So in our current society, at least in the developed world, reading is so fundamental that we try to make sure that every single kid learns how to read. However, unlike spoken language, which it appears that people can learn passively through just being in an environment where language is present, written language has to be taught. You remember being taught to read and write in school. You don't remember learning how to speak your first language. So we didn't evolve the sort of fact that we have to learn this stuff in school. We have to be explicitly taught it already suggests to us that it's not something that we evolved a specialized reading system for. Yet, when we look across people, across cultures, we find that there is specialized circuits that appear to be responsible for processing the written word. Specifically, there seems at the bottom of the brain, at the junction between the temporal and the occipital lobe, there's this little patch that seems to be specialized for reading in the brain. And it's specialized across cultures, and so it responds when you're reading an alphabetic language like French or a logographic language like Chinese. It actually doesn't even require visual input, so the same part of the brain responds when blind people are reading Braille, their written language. It seems to change with literacy, so as somebody becomes literate, you see that the response profile of this part of the brain changes where it's now reading specific. And maybe based on my own work, most strikingly, when you lose this region, you end up with selective problems in being able to read. And I just want to illustrate this last point with a case study because I think it can be hard to sort of grasp what it means to so selectively lose the ability to read. So I want to introduce you to the case of MTS. So MTS was 11 years old when, unfortunately, in 2011, she suffered a cerebral hemorrhage that left her completely unable to read. It was really selectively reading because other types of visual information seemed to be fine to her. So this is her copying a picture that's like a classic neuropsychological test of 2 trees, a fence, and a house. And you can see that she was able to see those objects just fine and copy them just fine. However, when she was shown just the simple shape and asked what she saw, she described it as a blur or a smudge, and this is what she copied. And so to show this in even more detail, and yet she was actually able to write the words just fine, but was unable to read back what she wrote. So here is just a video showing her just naming alphanumeric characters and other types of characters she's presented. She actually has this problem both with letters and numbers, but you'll see not with things that are visually very similar to that.
I can't tell that one.
All right.
What is?
You can go on.
No.
Okay.
No.
Okay, that's okay. There are going to be a lot of these you can't see.
Square.
Uh-huh.
The number sign thing?
Yes.
So
2011 was a little pre-Twitter.
no. An exclamation mark.
Uh-huh.
An arrow facing to the right. I can't see that one. That one.
So she has an
Oh, that one.
unbelievably selective problem in letter and number perception, despite, say, only 5 years of experience being somebody who is a reader. So becoming literate results in specialized brain systems for reading. Yet this brain specialization is caused by developing some cultural expertise, not by some type of deterministic genetics. But even still, it's more complicated. And that's because about 10 to 20% of learners, depending on who's estimating, struggle to learn to read. And so they, despite no other cognitive impairments that are obvious, despite having the ample educational opportunity, learning to read is really hard for them. And part of this problem means that they fail to develop the normal neural network for reading. Specifically, they don't develop that little spot on the bottom of the brain and the back of the brain that seems to be specialized for reading words. And this seems to have a pretty strong genetic component. So people have known since they first described dyslexia that that it seemed to go in families. And with twin studies, people have been able to estimate that approximately 30% of the population variance is explainable by something heritable, though what the exact gene is for dyslexia is not something that anyone has gotten even close to discovering. And so we're in this really complicated situation, right? So seems to be some genetic component that seems to be predictive of whether you're able to learn this cultural skill of being able to read. So there seems to be something there that can be explained potentially by nurture. And so maybe we can make sense of this when we start to think about the learning problem that is posed by learning to read. And so when we're learning to read, we have to learn how to map visual input onto language representations of pronunciation and of meaning. As a cognitive neuroscientist, we sort of assume that this is mediated by a representation of letter shape, a representation of letter that abstracts away from information about the idiosyncrasies of how that letter was presented. So it doesn't care about case, it doesn't care about font, it doesn't care about the size. You just sort of learn the letters in the word and the spellings of those words. And so the learning problem is, how do you learn to map from vision onto those letter identities? And how do you learn to map from letter identities onto sounds? You might think of this as phonics. And sequences of letter identities onto meanings. You might think of this as something like sight word reading. And so once you can think of the problem that way, you can imagine how in this interactive system, problems with different components of the system can actually lead you to have trouble learning how to read. And so it's probably the case that for most people who have dyslexia, there's some cause that has to do something with development of the language system. So there might be some genetic difference in the development of the language system that means that these individuals have impoverished phonological representations, difficulty representing the sounds of the words. It might also not be genetic. It might be because you were a kid who had a lot of ear infections, and so therefore when you were hearing a lot of spoken language, it sounded a little bit like you were underwater for a lot of your childhood. In that case, you're trying to learn the mapping from the spelling of a word onto the sounds, but you have uncertainty about what those sounds are, which makes learning this mapping really hard. Humans are really great at coming up with compensatory strategies, so it's possible that in your everyday life with spoken language, nobody would have ever noticed that this problem existed, and it isn't until you're in the classroom learning how to read that the subtle problem with phonology starts to come out because of the learning problem that gets posed. This isn't the only part of the system that could cause this problem. And so another way that you could imagine this is somebody who has some problem with their visual system. And so I'm going to talk to you about another case study of an individual who had a developmental problem that led her to have a form of dyslexia. And so this student was an 18-year-old student at Johns Hopkins University who came up to her college professor at the end of class one day to talk about her lifelong problems in writing and math, and to a lesser extent reading. As a result, I now ask all my cognitive neuroscience students if there's anything interesting going on with them, because who knows who's going to be in your classroom and what you could sort of learn from them. So the person who studied her, Mike McCloskey, who was one of my graduate advisors, spent 3 years looking at her spelling and reading and math problems before he came on what the ultimate cause of the problem was and wrote a whole book about her. So the ultimate cause of this problem had nothing to do with the reading system. It had to do with the visual system. She was unable to tell when she looked out at the world whether something was presented on the left or on the right, despite fully being able to tell what the object was. And so here's just an old video of her doing this, so I apologize for the graininess, but it's a very simple task. She just has to identify the object and then indicate where it is and then look at the object. You can't really see what this object is, but it is a cylinder, and she's going to get that correct.
Can I see a cylinder? Look at it.
Well, tell me which side.
Oh, it's over here.
Okay. Now look at it.
Okay, I see a cylinder.
So at this moment, she believes she's staring directly at a cylinder when, of course, she is not. It is reflected across her midpoint. She makes reflections across the midpoint, both left and right, and top and bottom. And so when we think about this in terms of the problem of learning to read, it means that she could never tell apart a lowercase b, d, p, or q. And she couldn't tell with any consistency the order of the letters in the words she was seeing, so she couldn't tell whether she was reading the word lemon or melon. And so as she was trying to develop these mappings between the visual input she was seeing in the world and the sort of language representations, she had all this uncertainty built into the system, so she was never able to fully learn those mappings. And so I'm just going to reiterate what I guess everybody has said, which is the relationship between nature and nurture is super difficult to untangle. The brain is shaped by experiences, and just because we see that everybody's brain is approximately the same doesn't mean that that's hard wiring. And so there's variation, there's probably genetic differences, but those genetic differences can have far-reaching implications that go well beyond just the system that's impaired with those or different because of those genetic variations. And I want to end with a note about why I think this really matters. People actually think that when we talk about differences in the brain, we're probably talking about things that are attributable to nature and not attributable to nurture. But as I said at the beginning of the talk, the brain is the organ that gives rise to all of our behavior, all of our thoughts, all of our feelings, our identity. If we think that those things can be shifted by experience, it better be the case that our brain also can be shifted by experience. And so think about all the things that you've ever heard about how men and women are different from each other. Men are from Mars and women are from Venus. It is almost certainly the case that much of that variation can be explained by social factors in how we raise boys and girls. There's wonderful work in developmental psychology showing that if you have adults interact with a baby that they believe is a boy, they will behave differently towards that baby than if it's the exact same baby that they are led to believe is a girl. Yet, as soon as you start to talk about these differences between men and women as being caused by something in the brain, as opposed to some behavioral difference, the way people start to attribute these things to nature versus nurture changes. And so we've actually done a study on this in the lab. It's a different kind of study than what we mostly do, but I think it's a really important point to make. And so we gave people passages of text that described some result that had some difference between men and women in it. Here is a classic example about visuospatial processing, or some type of thing that people like to make claims about. And then we ended those passages either with a statement that didn't have anything to do with neuroscience, or a statement that had a small number of things in it that had irrelevant neuroscience, just describing where in the brain this difference might have emerged. And we had people then rate these scientific explanations as to whether they thought they showed that there were essential differences, biological, nature-based differences between men and women, or whether the differences between men and women were caused by social factors. And not surprisingly, but depressingly, we found that just introducing irrelevant neuroscientific information made people think that these differences were more essential and less socially constructed than if we gave the exact same things without that irrelevant neuroscientific data. But of course, if there's one thing you take away from my talk, if experience can change your behavior, so can your brain. The brain does not have a privileged status in terms of talking about nature, whereas behavior is somehow about nurture. And so this is your brain. Your brain is amazing. It's the way that it's connected and the way that it works is caused by so many things. I can understand why people want a simple explanation as to what parts are caused by nature and what parts are caused by nurture, but we're never going to get that. And I don't think it's even that important, because I fundamentally agree with the thing that Caleb actually ended his talk with, which is what we do know is that there's no aspect of behavior that's simply caused by genetics. For any aspect of behavior, we know that there's things that we can do in the environment that can change those behaviors. And so that gives us the opportunity to enrich environments to make every human the best possible versions of themselves. And that's why I think we should probably put this debate to rest. So thank you very much. Oh, we're
Let's see if the sound's working. No, it's not. We have time for a few questions, so if somebody has a question to ask, I'll go ahead and point and address it anyway. Yes.
As you point out, it's unusual to say that behavior can be purely genetic, and every genetic behavior can be affected by the environment. The environment is doing everything, and we can make environment and we can change people as much as we want, as if that's true, when we know you can't change people as much as you want, right? There are biological limits, but you don't want to ever do that argument. Why not?
Well, so because I think that my perspective is more hopeful. So we don't
Say to me, yes,
Now it's become ridiculous thing.
you haven't met the requirement to be optimistic.
I think if we're trying to understand why humans the cause of trying to understand why humans behave is frequently to try to understand the ways that we can behave better. So instead of just sort of giving up on these kids who have phonological problems that are having trouble learning to read, instead can we think about what are the other kinds of environmental factors, what are the other kinds of learning, teaching tools we could design for them that deal with the sort of problems that they have so that they can become the best versions of themselves. I didn't actually say that anybody can become anything. I said we should try to figure out how to design environments to get people to be the best versions of themselves. Save, we're not going to be able to go in and change people's genes around, nor should we want to do that because of the crazy unintended consequences that could eventually come from that. But we can do things with people's environments to try to help them become better versions of themselves. And so that's why I think it's sort of a fundamentally optimistic.
Yes, it is true that we are constrained by our biology. We can't breathe underwater. However, I think in any, I'll make a really strong statement, any situation where you're making a measurement and you want to ask the question, can I change an individual so that they will be better than this measurement? I think the answer is yes. We can manipulate anything. So even if you want to change your height, I can probably find some cocktail drugs that will cause you to grow a small amount of height, right? So yes, there's things that are locked in biology that are very difficult. But if we focus on it, we can find it. So to say that we should not, I guess the implication of Simon's statement is it is worthwhile studying because we can find the ways that we can help people. And I don't know the implication of the opposite statement, that nature constrains us, I don't know what its implication is.
I'm just going to let Julia jump in if she wants to. In this group, go ahead.
I mean, as someone who works on flies, so I don't have any hope. I don't care if there's hope or not. I mean, I think it's an open question. And certainly if you think over longer evolutionary timescales, it's really an active area of research of like, how, can we evolve pigs that can fly? Like, we don't know. I will add one more thing, which is that the proportion of phenotypic variance that's attributable to genetic variance, so heritability, is itself shaped by the environment. So, for example, educational attainment in humans is more heritable among individuals who are in a higher socioeconomic status. So the idea that, like, even that it's both, and there's constraints and there's environmental enrichment, that itself can be variable and change. So I don't know. But
How many male fruit flies have you seen that can lay eggs?
None, because the definition of biological sex is based on gamete size. So the definition of biological sex is the relative size of the gametes you produce. And so if you lay eggs, by definition, you're a female.
The point is that Martin is right. You guys are somehow trying to simplify one simplistic picture all the way to the other side to another simplistic picture. And instead of giving us really something which you say, there are, part of the issue, for example, the elephant in the room is we want to understand between the male and female behavior. Okay? And we can say we don't yet fully know. We can say what we know, we don't know. But to pretend that we know that overall there are differences. And the question we don't understand, what are the behavioral differences? So it's okay to say we don't know, but to pretend no, they are the same, everything is behavior. I don't think that's what science is, and I'm sorry.
I don't think any of us said that everything is behavior.
No, you're already implying, this is the direction you're already implying. There is an undertone.
So
It
what
was
was
Martin
really
and I
negotiating is that elephant appeared suddenly in my mind. It
Yes,
wasn't actually
actually,
here.
this is the other side.
It wasn't so for me.
Before the elephant.
Yeah, no, I mean, I think that I will stand by my statement, which is that the problem that you perceive is a problem of poor definition. So you made a tautological definition, right? So you said laying eggs, but that's typically not the thing that we care about. And
Speak for yourself.
in the lab, we care very much about that. I guess my point is, but I'm assuming that that's not actually your concern. Your concern is not that men don't lay enough eggs. And so I guess the argument is that when you refine your question to be an interesting question, we find that the environment has incredibly strong effects.
I think what I'm going to do in this environment is thank the speakers and then take this outside in separate quarters. It sounds like there's some very fruitful discussion going on. So thank you very